Hello folks, and welcome to video number 2 on cell division. Now, the ability of organisms to reproduce more of their own kind is just one of the characteristics that distinguishes living things from non-living things. Rudolf Virchow, a German physician in 1855, put it best when he said, "Where a cell exists, there must have been a pre-existing cell." He summarized this concept with the Latin axiom, omnis cella e cella, every cell from a cell. For prokaryotic cells like bacteria, the process appears simple. As the individual bacterium grows, it responds to either internal or external signals to prepare for cell division. The process in prokaryotes is called binary fission. The bacterium copies its loop of DNA, its chromosome, and the membrane and the cell begin to pinch off. The two new cells are referred to as daughter cells. Really, there's no gender to these cells. Biologists simply refer to them as this because these cells have the ability to divide and produce more cells. So we sort of improperly use the term mother cell and daughter cells. One thing to note here is that the daughter cells are genetically identical to one another. In a unicellular organism, like this eukaryotic amoeba, cell division reproduces the entire organism. Each generation is identical to the next, as the chromosomes are copied and passed on faithfully. Imagine if there were this many of me. In multicellular organisms, cell division is necessary for growth and development, as well as repair of old or damaged tissues, as seen here, as a tree grows new tissue over a damaged portion of the tree. Your tissues are capable of the same thing. Eukaryotic cell division of somatic, or body cells, is a complex process that involves a cell cycle. This is the growth of the cell and the process that copies its genetic material or chromosomes. Mitosis, this is the division of the nucleus, followed by cytokinesis, the division of the cytoplasm. The cell cycle or life cycle of a eukaryotic cell consists of a mitotic phase or mitosis that's followed by cytokinesis, and then interphase. This is when the cell is actually doing whatever job it was developed for. If it's a white blood cell, it's involved in immunity. A brain cell, it's sending and receiving messages, or a skin cell that may simply be covering a body surface. Cells spend most of their time in interphase. But interphase is a bit more dynamic than just having a cell do its job. There are distinct changes that can occur inside the nucleus and the cytoplasm that has caused us to divide interphase into 3 distinct phases. Gap 1, or the G1 phase, when the cell simply grows in size. The synthesis, or the S phase, when the cell makes copies of its chromosomes. And Gap 2, or G2 phase, when the cell prepares for cell division by replicating its organelles. We can create a sort of cell cycle clock that shows the phases of the cell cycle. Interphase takes up most of the clock time, while mitosis and cytokinesis are short periods of time. Depending on the type of tissue a cell belongs to, the time it takes a cell to go through the cell cycle varies. Skin and bone marrow cells divide often, especially when we're growing or if tissue damage requires repair. Some cells, like nerve cells, enter the G1 phase and never move on to divide again. In a sense, their clock stops here at the G1 phase. They enter into what is called the G0 phase and never progress past that. This is why damage to nerve tissue is typically not reversible. The cell cycle is carefully controlled by external and internal signals, and we'll talk about these in part 3 of cell division. But let's focus briefly on the events of mitosis. A brand new cell starts its life with its complete set of chromosomes. Here, we'll keep it simple and imagine the new cell with just one set of chromosomes. While in the gap 1 phase of the cell cycle, the chromosomes, made of DNA, are these singular structures. As the cell enters the S phase of the cell cycle, it it has copied each chromosome. The two halves, called sister chromatids, are identical copies of each other. The cell is now ready for the M phase, or mitosis. In animal cells, these structures, called spindle fibers, emerge from the centromeres. The sister chromatids separate and move along the spindle fibers to opposite ends of the elongating cell. Cytokinesis begins and the membrane pinches off, finally producing two new identical daughter cells. The term diploid is used to describe the fact that each cell still has the complete set of chromosomes. At some point in their life cycle, the cells will have both sets of chromosomes and are referred to as diploid. In some cases, the cells only have one set of chromosomes and are referred to as haploid. Here's a series of photos and illustrations of a cell going through the process of mitosis. These pictures here are neat. They're actually photos of cells using a staining technique called fluorescence, which causes features to glow in different colors. Starting here with a cell in interphase, the nucleus is intact with its nuclear membrane. The chromosomes are unwound in a form called chromatin. The cell is going through its business, doing the job it's supposed to do. If it gets a signal to divide, it will copy each of its chromosomes and enter into M phase, that is, mitosis. We divide mitosis into 4 of 5 phases that involve distinct events in the cell cycle that mostly involve the nucleus. The first phase is called prophase. Here, the nuclear membrane begins to disintegrate. The chromatin begins to condense into their more visible chromosome structures. Meanwhile, the centrosomes begin to migrate to opposite ends of the cells and form an array of microtubules called the mitotic spindle. In pre-metaphase, the chromosomes attach to an individual spindle fiber at their centromeres, at points called the kinetochore proteins. They're located on either side of the centromere seen here. Each fiber is now called a kinetochore fiber. At metaphase, the spindle apparatus then arranges each chromosome along the equator of the cell, sometimes called the metaphase plate. You can see some of the fibers from the spindle apparatus are not connected to a chromosome. These are called non-kinetochore fibers and will continue to grow in length. These are responsible for causing the cell to elongate. In anaphase, each kinetochore protein starts their way along the kinetochore fiber toward opposite ends of the cell. This causes the sister chromatids to separate. Each chromatid thus becomes a full-fledged chromosome. The cell continues to elongate, and the last step in mitosis is telophase. Here, 2 new nuclei begin to form, and the chromosomes unwind back into chromatin. Then cytoplasmic division, or cytokinesis, begins. In animal cells, a cleavage furrow appears and continues until one cell becomes 2. Each daughter cell has identical chromosomes or genetic material in their nucleus. Plant and animal cells both grow through mitosis in a very similar fashion. One difference is that the spindle apparatus doesn't appear in plant cells. And when cytokinesis occurs, instead of pinching off, plant cells form a new cell wall between the 2 nuclei. I think it would be a good idea to watch this animation of mitosis that I found in a McGraw-Hill website. Pay attention to this.
During interphase of the cell cycle, the genetic material of the cell is found in the form of chromatin and located within the nucleus of the cell, which is surrounded by the nuclear envelope. During the S stage of interphase, DNA replication takes place, and thus the cell leaving interphase and entering mitosis already has replicated its genetic information. In addition to the replicated genetic information, interphase cells that are about to divide also replicate their centrosomes. In animal cells, these centrosomes are formed from centriole pairs. During interphase of the cell cycle, the genetic material of the cell is found in the form of chromatin and located within the nucleus of the cell, which is surrounded by the nuclear envelope. During the S stage of interphase, DNA replication takes place, and thus the cell leaving interphase and entering mitosis already has replicated its genetic information. In addition to the replicated genetic information, interphase cells that are about to divide also replicate their centrosomes. In animal cells, these centrosomes are formed from centriole pairs. As cells leave interphase and begin prophase, the first stage of mitosis, the chromosomes begin to condense. The nuclear envelope begins to vesiculate, and the centrosomes migrate to opposite ends of the cells to define the two poles. During prophase, the mitotic spindle apparatus begins to form by the polymerization of tubulin proteins. This creates microtubules that emanate from each centrosome. Chromosomes completely condense such that sister chromatids formed from the replicated genetic information and joined at their centromeres are clearly visible. During prometaphase, the nuclear envelope completely breaks down and the mitotic spindle is formed. Polar microtubules extend from both poles and overlap with each other. The sister chromatids become attached to the mitotic spindle via kinetochore microtubules that attach to the kinetochores, which are bound to the centromeres. As prometaphase is nearing its end, the sister chromatids are observed to jerk back and forth between the two poles. During metaphase, the sister chromatids become localized in a region called the metaphase plate. Once the chromosomes have aligned at the metaphase plate, metaphase is complete. When metaphase is completed, anaphase begins. The sister chromatids separate, and each chromatid is now linked to only one pole via a kinetochore microtubule. The kinetochore microtubules shorten, and the individual chromatids, now called chromosomes, are drawn to the poles. Toward the end of anaphase, the polar microtubules push against each other and cause the two poles to be moved farther away from each other. Each daughter cell will receive the same complement of chromosomes as was originally found in the mother cell, thus assuring that each daughter cell gets an identical copy of genetic information. During telophase, the chromosomes decondense and most of the microtubules depolymerize. Portions of nuclear membrane surround each individual chromosome, creating vesicles. The vesicles fuse with each other to create a nucleus containing all of the chromosomes at one pole of the cell. In animal cells, cytokinesis, or cell division, involves the formation of a cleavage furrow. The plasma membrane is observed to constrict and eventually separates the cell into two daughter cells. The daughter cells then enter interphase, where growth and enlargement occurs, leading to either preparation for another mitosis or differentiation and specialization occurs.
That's it for this video for now, but I want you to remember that mitosis is important for at least three things. Number one is for the reproduction of unicellular organisms, but for multicellular organisms, it's necessary for growth and also repair of damaged tissues. I hope that video was useful. If you have any questions, write them down and bring them to class. Until then, be well.